Guys, now let's talk about some of the most intricate and very complex machinery, not only in cells. I mean, whatever we know about universe until now, perhaps the machinery we are going to talk about is the most complex one in the whole universe, and that is the electron transport chain. As we have already told you that electron transport chain occurs in mitochondria, which we rightly say the powerhouse of the cell. This electron transport chain starts with taking electrons from two very important molecules, NADH and FADH2. These are actually the electron donors. They carry the electron and they give the electrons to different complexes of the electron transport chain. From where the NADH and FADH2 comes? They comes from the different metabolic pathways like Krebs cycle, like glycolysis. And the electrons once come in the electron transport chain, they pass through different carriers. We will go into mechanistic detail of this in the succeeding topics, and then the energy is used to pump the hydrogen ions, because the energy which is produced because of this transportation of the electrons. Protons are actually involved in the synthesis of ATP, and these protons gets into a gradient. A gradient is formed in the electron transport chain through the action of electron transport chain, which gives rise to the formation of ATP. Again, we will talk mechanistically about the synthesis of ATP through protons later. Oxygen is the final carrier of the electrons in electron transport chain. The presence of oxygen actually is the reason that we also term whatever is happening through the electron transport chain as the oxidative phosphorylation. The ATP formation through the electron transport chain, because of the presence of oxygen, is called as oxidative phosphorylation. क्या लर्न करेंगे हम आने वाले टॉपिक्स में? First of all, we will talk about different components of electron transport chain. We will see how the transport chain is coupled with the synthesis of ATP. We will see how different inhibitors can be used to really inhibit the performance of ETC, the working of ETC, and how the ETC is regulated.